Namaskar and welcome to CEC Gurukul lectures. I am Dr. Kumar Shantanu, Associate Professor of Botany from Deshbandhu College, University of Delhi. In this series of lectures on analytical techniques in plant biology, we have started our journey with a basic understanding of the most common and most widely used tool of biological sciences, that is microscopes. We shall continue our journey with microscopes for a longer time because microscopes, with the advent of and advances in sciences, have evolved across the time period. In our previous lecture, we have tried to understand the basic principles of microscopy. What are basic differences between light microscopes and electron microscopes? While understanding the basic principles and different types of microscopes, we also went across the various structures of a typical bright field light microscope. Today we shall continue our discussion and we shall try to understand that various deviations or various variations of light microscope, how they work and how they help in understanding the microstructures of any biological object in a more resolute manner. So, as far as light microscope is concerned, a very basic variation from the bright field microscope or the light microscope is also referred to as the stereo microscope. The stereo microscope is used for the observation of surfaces of large specimens. Stereo microscope is also used primarily when three-dimensional information is required, like for the screening through the vials of fruit flies or going through the basic segregation of very small plant structures like mosses from a group of different species. These type of microscopes are very useful for micro manipulations also and dissections also, where the ability of wide field of view and ability to zoom in and zoom out in magnification is very, very significant. When we use stereo microscopes, we can work on live organisms or we can work on dead organisms also. So here in the picture you can see a typical stereo microscope. Let us scan through various structures and parts of a stereo microscope. To begin from the bottom towards the top, the lowermost portion on which the entire optical structure is supported is called as base. On this base, there is a source of illumination coming from this hole, and inside this hole resides an illuminating source. It could be LED lamp or it could be neon or halogen lamp. Now above this illuminating source, there is a transparent glass which is called as stage plate. On this stage plate only, the live or dead large sized objects are placed, and sometimes when these objects are placed on a slide, to hold the slide on a static position, the stage clips are used. So these are the two stage clips which are used to fix the slides on which the objects are kept. Now on one end of this entire base lies a long sturdy structure, and generally it is made up of metals, that is called as arm. On this arm, the entire setup of optical lenses is framed. This arm has structures which bears zoom knob, focus knob and the entire optical structure. the entire optical structure has different components of it. So one component happens to be the top light. So in some of the microscope, the light is thrown from above, and in some the light is given from below. Above this top light lies objective lens. Then there is a set of optical structures which gives it a stereo effect, and this is called as stereo head. On this stereo head, two ocular lenses or eyepieces are fixed. These eyepieces can adjust themselves in terms of diopter, and it can also move. The distance between these two eyepieces can also vary, which is called as interpupillary adjustments. So overall, a stereo microscope is observed with the help of both the eyes, and because of the two eyepieces, we do get a three-dimensional structural effect of the objects which we are observing. Here are some examples. Here is a large structure of a moss or a small leaf, and here a plant structure which can be seen through a stereo microscope. Let us try to understand what is the basic difference of a compound microscope and a stereo microscope. As we have already discussed, that in stereo microscope we do get a three-dimensional structure. That means we do get a perception of depth also. So here the similar object is once seen with a common compound microscope, and when it is seen through a stereo microscope, the detailed structure with a perception of depth and thickness is also seen. So we do get a better clarity when we observe any object through a stereo microscope. Now, many configurations of the light microscope have been added over the years, and specifically to add contrast to the final image. So the entire purpose of using a microscope is to understand the details of an objective in much more detail, and also this can be achieved with the help of additional contrast to the object, to the image which we are seeing. Now there are different types of variations. One such variation happens to be changing the phase of the light, and because of the change in the light, the contrast of the image is greatly enhanced. So we shall continue our discussion from stereo microscope towards a next variation of the microscope, that is called as phase contrast microscope. So what is a phase contrast microscope, and or what is phase contrast microscopy? So what has happened is, improved contrast with transparent cells can be gained by closing to an extent the condenser diaphragm on a bright field microscope. So when we gradually close the condenser diaphragm, we generally see that the contrast of the transparent cells enhances. But this closing of the condenser diaphragm is limited to an extent, because beyond a certain point it will start reducing the numerical aperture of the lens system, and thus the resolution of the object gradually starts decreasing. And to avoid all this kind of limitations, the research microscopes can be converted to phase contrast microscope with the help of different lenses and different components, which is part of phase contrast optics. So what happens in a phase contrast microscope? So a phase contrast microscope uses two additional components. One component is called as the condenser annulus. The other component is called as phase plate. So in a usual common bright field microscope, two additional components are added. that is condenser annulus, which is used along with the condenser lens, as the name suggests, and the other one is called as the phase plate, which is used along with the objective lens. So with the interplay of these two components, the phase of the incident light is changed, and because of this changing phase of the incident light, the image which is formed gives us much greater clarity in terms of the detailed structure. Now, using these two components allows the change in the phase of the light, and with the help of phase contrast microscope, the detection of different components of cells by virtue of slight variation in their refractive indices are achieved. By this method, specimens mounted in their appropriate medium can be observed directly. So, this allows visualization of not only the outline of the cell, but also many of its organelles and their arrangements. It is used for examining living unstained cells because biological materials almost inevitably diffract light. So here you can see clear difference between the two images formed. One is taken through a phase contrast microscope, and the other one is taken through a compound microscope. So when we use two components of phase contrast microscope, the clarity of the outline and also the intracellular structure gets enhanced tremendously. Similarly, the microorganisms can be seen with a much greater clarity when we use the principles of phase contrast microscopy. So, phase contrast microscopes, they use dark field illumination. So, what is a dark field illumination? So, dark field illumination produces image of brightly illuminated objects on a dark or black background. This technique has conventionally been used for viewing the object outlines in liquid media, such as for a quick check of the status of the biochemical preparation, or for cells growing in a tissue culture medium. So, how this technique is different from bright field microscopy? In case of bright field microscopy, the background is always white, whereas in case of dark field illumination, the background happens to be dark. For lower magnifications, a simple dark field setting on the condenser is sufficient. So how it is done? So there is a component which is called as phase contrast annulus or phase annulus, which is used with the condenser. However, for higher magnifications and for more critical dark field imaging, a dark field condenser along with a dark field objective lens is also required. So here you can see that for a dark field microscopy, here is your light source and here is your condenser lens. So along with the condenser lens, the components of phase contrast components are used, and when this condensed light passes through the specimen, there is a objective lens, and along with the objective lens there is another phase plate, and because of the interplay of these two components, one with the objective and the other one with the condenser, the background is dark, and in this dark background the images illuminate with its borders. Now let us try to understand the basic principle of a phase contrast microscope. So what happens is, for examining cells through bright field microscopy, cells are often killed, sliced into thin sections and stained. Although such procedures are useful for examining the details of cell's internal architecture or the anatomy of the cells or the intracellular components of the cell. However, with an increased emphasis on the study of cell dynamics, and especially in the field of motility, in the field of cell division and cell death, the techniques that allow visualization of intact or still living cells have become increasingly important. So, the bright field microscope generally uses the object when it is killed or sliced or it is stained. But as far as the dynamics of any cell or the division of any cell is concerned, when we want to observe them, we need the cells to be intact and living. So in such conditions, phase contrast microscopy is of immense use. The phase contrast microscope improves the contrast by exploiting differences in the thickness and refractive index of various regions of the cell. It is a form of interferometry whereby contrast is derived from refractive elements within the sample, which cause phase shifts in light that are subsequently converted into amplitude changes which are registered as light or dark regions against a uniform reference. Let us try to understand the working principle of phase contrast microscopy with the help of a diagram. So, this diagram illustrates the pathway of light. The color symbolizes different types of lights. So, here is your scattered light with a 90-degree phase shift, and here is the background light with a minus 90-degree phase shift. So when light starts coming from a light source, the condenser, with the help of condenser annulus, it changes the phase of a light to an extent. And when it indices on a specimen or on an object, based on the thickness of the specimen or based on the internal organization of the specimen changes the phase to some extent. And this changed extent in the phase of light which is passing through the specimen is further focused with the help of the objective lens. This objective lens again has a phase plate attached to it. This phase plate attached to it restricts certain background phase of the light, whereas it allows only those phase of the light wavelength to pass through which has been changed by the specimen. Thus, the image which is formed at the image plane is the resultant of those shifted phases of the light which is caused by the specimen here, and thus the outline of the object is very, very clearly observable. So what happens is the cell organelles are made up of different proportions of various molecules like DNA, RNA, protein, lipids, carbohydrates, salts and water, etc. The regions of different composition have different refractive indices. As the rays pass from the light source through the object, their velocity might be affected by the physical property of the specimen. And inside the specimen, their velocity is also affected differently by different components of the cell. For example, when the light passes through a DNA or RNA molecule, the velocity is affected in a different manner when as compared to when light is passing through a protein or a lipid or a for say carbohydrate molecule. And along its route, the velocity of the rays slows down to varying extent upon encountering different regions of the specimen. So in a phase contrast microscope, we exploit this change of velocity which happens at varying extent. So light passing through the thicker parts of the cell is held up relative to the light that passes through thinner parts of the cytoplasm. This results in a change in phase related to the light wave that have not passed through the specimen. Although such changes in phase are not detected by the human eye, the phase contrast microscope overcomes this problem by converting these slightest differences in refractive indices into differences in the intensity. This differences in the intensity is converted or is changed into relative brightness and darkness, and this change in this differences in intensity, which is reflected in terms of brightness or darkness, is now further visible to the eye. So the slightest of the change in the phase of the light, which was earlier not perceptible to the human eye, is now visible to the eye because of the two components which we have inserted in a phase contrast microscope. So here this entire thing is represented with the help of a diagram. So what happens here is there is a light source which passes, and when it passes through an object, the velocity of this middle light component changes, it slows down, whereas those light waves which are not passing through the object keeps on moving at their common or at their usual speed. So the wavelength of the light, or the light component, or the photons which passed through the specimen, because of the thickness of this specimen and because of the intracellular components, the speed of the velocity of this photon slows down, and this creates a change in the phase, and this change in the phase is perceived, it is perceived at the image plane. So this is how any change in the slightest amount of the velocity is further enhanced, and it is made visible in terms of brightness or darkness because of the components of the condenser and the components of objective lens, that is phase plate. And when we observe it through our naked eye, we can see the change in brightness and darkness, and the contrast is thus further enhanced. So a phase contrast microscope accomplishes this result by two steps. First, it separates the direct light that enters the objective lens from the diffracted light emanating from the specimen, and by causing these two types of light rays to interfere with one another, producing an image by highly contrasting bright and dark areas against a uniformly illuminated background. So on an average, light passes through the transparent specimen is retarded by approximately 1/4 wavelength, and the direct undifracted light passes through a portion of the phase plate that speeds it up by approximately quarter wavelength. So this can be understood, or this can be observed with the help of this diagram, where any light which passes through the specimen through the phase plate, it changes the wavelength of the light. So phase contrast microscope is equipped with phase lenses and a corresponding phase ring, that is called as annulus in the condenser, as we have already discussed. So this conversion from a bright field microscope to phase contrast microscope is accomplished using a phase plate. The phase lenses contain a phase plate that works in tandem with the phase ring to retard direct light by 1/4 of the wavelength. So these are the two components which we have already discussed, that is condenser annulus and the phase plate. So light that passes through the specimen will be diffracted by organelles, cell margins, etc., and this light is retarded in phase. Background light and light that has passed through the specimen interact at the image plane, causing wave interference with objects appearing bright against a uniform background. So such variations are done to enhance the contrast of any common compound microscope. We shall try to understand another variation of microscope, that is called as polar light microscopy, in our next lecture. Thank you so much for your patient listening.